Good morning, everyone, and welcome to the 2018 Royal Tyrrell Museum Speaker Series. Today, the Royal Tyrrell Museum and its cooperating society are proud to present our very own Dr. Caleb Brown. Caleb is the Betsy Nichols Postdoctoral Fellow here at the Royal Tyrrell Museum. Caleb is originally from northern British Columbia but grew up in Red Deer, Alberta. He received his bachelor's degree in zoology from the Department of Biological Sciences at the University of Calgary and spent 3 summers during his undergraduate years working in the preparation lab here at the Royal Tyrrell Museum. After completing his bachelor's degree, Caleb stayed at the U of C to pursue his master's degree, again in the Department of Biological Sciences, where he investigated the diversity of small plant-eating dinosaurs in the late Cretaceous of Canada. After successfully defending his thesis, Caleb moved out east to pursue a PhD in the Department of Ecology and Evolutionary Biology at the University of Toronto. For his dissertation, he studied the variation and evolutionary morphology in horned dinosaurs from Dinosaur Provincial Park. Fresh out of his PhD, Caleb moved to Drumheller to take on a postdoctoral fellow position here at the museum. Caleb's research interests focused primarily on the evolution of dinosaurs, particularly of ceratopsians. Over the years, Caleb has conducted fieldwork in Alberta, Saskatchewan, the Northwest Territories, China, and Mongolia. Today, Caleb will give a talk about the world's best preserved Ankylosaur borealopelta and what the new information about this specimen has revealed about armored dinosaurs. So without further delay, I present you Dr. Caleb Brown.
Can you all hear me at the back? Okay. Thanks, Francois. Thanks for all coming this morning. First off, I have to apologize because I am dealing with a bit of a cold, so hopefully my voice will hold out for the whole talk. In spite of that, I'm actually very excited to be here because I get to talk about this specimen, which is by far the coolest specimen I've ever been involved with researching, and probably the coolest I will ever be able to be involved with researching. And it is the exceptionally preserved nodosaur that our museum announced to the public in the summer, just last summer. And what I hope to do with this talk is give you a bit of an introduction into the specimen. Some of you already know the story, but about the discovery, the collection, preparation, and then talk a bit about why it's unique and highlight some of the research that's been done so far. And that's just the tip of the iceberg because there'll be a lot more research done on this specimen as it progresses. The first thing that's I need to mention about this animal is that its discovery was actually quite miraculous. And that's because it was discovered by this machine here, this excavator, in the oil sands mine, Suncor Millennium Mine north of Fort McMurray. And you don't want to get a good sense of scale in this image. Here's a sense of scale with a person. These are gigantic excavators. The operator will be standing almost 10 meters above the ground. And each bucket full of rock that it picks up can contain up to 44 cubic meters of rock. So the fact that this is what hit the dinosaur and the operator noticed it, we need to be give proper attributes to the discoverer. And that was Sean Funk. He was the operator of this equipment that day. And on the afternoon of March 21st, back in 2011, he was doing overburden removal. So he was digging through the rock down to the productive layer, the layer that has the petroleum in it. And he noticed that he hit some rock that was unusually hard. It was much harder than all the surrounding rock. And him and his supervisor, Mike Graton, stopped and they saw these kind of polka dot-shaped rocks coming out of the hill. And this is what they found, posing happily next to their find. And instantly they knew this was something important and needed to be reported. Now, the fossil laws in Alberta are some of the most progressive in the world, in that any industrial mining in areas that have paleontological resources, there needs to be monitoring. And if anything important is found, it needs to be reported to the proper authorities, which in this case is the Royal Tyrrell Museum. And then we respond, we assess the find, and try to collect it as quickly as possible. And that was the case here. With Suncor's help, we had a team on the ground in a couple of days, and that team was led by Donald Henderson, our curator of dinosaurs, and Darren Tanke, one of our senior technicians. And their job was to assess the find, figure out what it was, and the best way to collect it. Now, before I can go any further, I need to give you a bit of context, because the rock that they're digging through in this mine is actually marine rock. Back in the Cretaceous, there was a large inland sea covering most or the central part of North America. And the rock that they're digging through is called the Clearwater Formation. And it was deposited not in rivers or flooding events that we see the rock around Drumheller, but it was deposited out in the marine shores. It's actually also much older. It's about 40 million years older than the rocks in the Drumheller area or the rocks down at Dinosaur Provincial Park. Because it's a marine environment, we would expect to find marine animals, and that's the case. Most other stuff that's been found up there, and we have got a lot of other exquisite animals, have been marine animals. So here on the top we have Nicholsaura, which is a plesiosaur, and on the bottom we have Athabascasaurus, which is an ichthyosaur. So this was kind of the default assumption going in, that what we had here was probably another marine reptile. That changed fairly quickly on the ground with the notice that we had actually preserved osteoderms. This is armor that's preserved in situ, in place for the animal. And because there's armor, it actually really limits the amount of possibilities in terms of who could or what could be preserved in terms of the skeleton. What an osteoderm is it's basically a bone embedded in the skin. It basically means skin bones. And there's a lot of animals alive today that have osteoderms, things like armadillos, crocodiles, various lizards. And there's also a variety of things that were extinct that had osteoderms as well. Just to show you what this looks like in a more internal sense. So here are some amazing CT scans provided by Ed Stanley at the Florida Museum. And on the left, you have the animal as it was in life, and on the right, you have a CT scan. So everything but the bones has been removed. And he's kind of color-coded it. So all the stuff that's tan, that's the internal skeleton. That's the skeleton that we have. And all the green stuff is the osteoderm. So those are separate bones embedded in the skin. And the big take-home message here is that those bones aren't connected to the rest of the skeleton. They're kind of loose. They're held in that skin. They don't actually connect to the internal skeleton. The best example of osteoderms, or one of the best examples we have today, are living crocodilians. And the picture on the top, you can see what the skin looks like while the animal's alive. It's covered in this kind of leathery skin, but within that skin you have these bits of bone that form this network that cover the animal's back and the sides and the belly. So given that we have these osteoderms and that they are fairly widely distributed in these distinct rows, we knew that this was from what's called a Thyreophoran, a armored dinosaur. This is the group that includes things like stegosaurs and ankylosaurs. And they're characterized by their armor. They have kind of two different skeletons. They have an internal skeleton, or endoskeleton, shown in blue. And this is the same skeleton that we have. The bones are the same, they're just in different numbers or different proportions and sizes. But uniquely, they also have what's called an exoskeleton or dermal skeleton. And that's formed by these osteoderms, bones embedded in the skin. And this is much rarer in terms of animals that possess osteoderms. And if we look at the pattern of evolution of the dermal armor in the armored dinosaurs, we see a very general pattern where the earliest forms have kind of simple, small armor spread throughout the body. One group then has the armor restricted to a pair of rows that run down the back and modified into either plates or spines. And a good example of this is Stegosaurus, and this is the group called Stegosauria. Then there's another majoration called Ankylosauria, and this includes both Ankylosauridae and Nodosauridae. And they have osteoderms widely distributed along their body, but they tend to have them in very distinct transverse rows, especially around the neck. And Ankylosauridae specifically has a tail club that's made up of osteoderms at the tip of the tail, whereas Nodosauridae does not have a tail club, and instead they have large shields, or spines coming off the shoulder region. This is one of the areas that gets confusing because the term ankylosaur can mean two different things, or multiple different things. It can mean Ankylosauria, so that entire group, or it can often mean just Ankylosauridae specifically. So when I'm saying ankylosaur, I mean Ankylosauria, the group that includes both Ankylosauridae and Nodosauridae. And fairly quickly, it became obvious that what we had here was an important specimen and an exceptional specimen for a number of reasons. First off, all those osteoderms, they're still in place. They're still in the original pattern. And the endoskeleton, here we have some vertebrae preserved and some tendons, they're also in the original position. This is what we call articulated. And this is important because it means that the bones are still in the life position. They're not all jumbled up. So we can learn a lot more about the anatomy when everything is still in place. But we didn't just have the bones preserved. There was other stuff preserved as well. You can see these diamond or octagonal shaped black areas or dark gray areas. This looks like it might be preserved skin associated with those osteoderms. And there's also these kind of amorphous blobs of gray with these orange spheres. And this might be stomach contents. So already we have an articulated dinosaur, possible skin, possible gut contents. So it was something that was definitely a high priority, important specimen. But we didn't really know how complete it was. Those blocks that I showed you initially, those come from the hip region area called the sacrum. And that's what had fallen out of the cliff and had been noticed by Sean. But half of the skeleton was still up high in the cliff. We didn't, you can see a cross section here showing kind of the bones still coming out of that cliff. But it wasn't clear which half was still in the cliff. Did we have the top situation, where the front end was going in and the skull was still probably there, or the bottom situation where the tail was in the cliff and the head had already been hit? And hopefully, of course, we have the top situation, and as many of you already know, that is the case. We do have the front half of the animal preserved, which is much better than having the back end preserved. So in terms of a collection, the first stage was kind of a salvage. Because it had been hit by the excavator, there was chunks all over the place. So a couple of days were spent collecting everything off the ground in hopes of putting this back together, back at the museum and putting the animal back together. The second stage of the excavation was getting down to the part that was still in the rock. And that was done largely with the help of Suncor and the big equipment that they had on site. They both dug down to the level of the fossil the fossil in the cliff, and also using that rock to create a ramp for easy access from below. The idea was to isolate the skeleton into one large block, dig underneath that, coat it in burlap and plaster and cement, and then try to lift it out onto an awaiting a truck. Unfortunately, as many of you know, there was a bit of a catastrophic failure. The jacket didn't hold, and it broke into two big chunks and a whole bunch of little chunks. So again, it was back to kind of a salvage situation. Pick up all the pieces, bring them back to the museum, and put them back together again. But despite this, a couple weeks later, all of the chunks arrive here at the museum and are offloaded back into our collections facility ready for preparation. And this isn't the end of the kind of first part. This is actually just the beginning, because the next step is the preparation. And the specimen was handed off to the very capable hands of Mark Mitchell, who's one of our long-term technicians. And Mark's job was to slowly remove all of the rock down to the surface of the specimen, consolidate everything, glue it back together, and put everything back in the original position. It was no easy task. Here are some just photos showing the progression for that skull. Excavating it from the rock, exposing more and more, and getting it ready for research and display. Now, Mark started the preparation in April of 2011, and he didn't work on much else other than this, and he didn't complete it until December of 2016. So it took about 5.5 years of dedicated work, about 7,000 hours, basically just working on this specimen alone. And the reason it took so long is a couple of factors. First off, the size. The bigger the specimen, the more time you need to put in to prepare it and get it ready for research. Secondly, the rock was very hard, and although the specimen was incredibly well preserved, it was very soft. Mark describes it as kind of compressed talcum powder. So he was fighting millimeter by millimeter to expose it without damaging the underlying fossil. And finally, normally during preparation, you're removing rock down to a bone surface, and bone has a very discrete outer surface. In this case, on top of the bone, there was often skin or some other soft tissue, which is much less obvious, much more discreet. So you have to be much more careful not to remove that thin organic lining. Excuse me. But after 5.5 years, what emerged was one of the best preserved dinosaurs ever found. I'm sure many of you have seen it. If you haven't, you have to go see it in the galleries. It is, without a doubt, a world-class specimen. And there's one reason that makes it so distinct, that makes it unique from all the other dinosaurs that have been found. And that's the combination that almost the entire surface is covered in preserved skin, which means you can't actually see the skeleton underneath. And excuse me, preserved dinosaur skin is not rare. We do find lots of patches. Sometimes we have fairly large patches. But when we have skin preserved, often the animal is kind of flattened like a pancake. It looks like roadkill, or it looks like a dried up piece of beef jerky. In this case, that's not the situation. The animal retains its three-dimensional shape. So it looks now, as it's preserved, almost exactly the same as it looked back in the Cretaceous, which is quite a thing to think about. It's hard to over describe how well preserved this is. These are some great photos from Robert Clark from National Geographic. And I just want to highlight some of the really cool features, really distinct features about this animal. First off, it has this very triangular skull. This is a nodosaur characteristic. Ankylosaurs tend to have very short, rounded skulls, so instantly we know that this is a nodosaur. It has these three large rows of osteoderms on its neck with big keels, and those osteoderms also continue all the way down the back. And then in the shoulder region, it has this large spine called a parascapular spine kind of sticking out from the side. There's a few other more subtle things in the animal. If you haven't seen these, go check it out in the gallery after. On the animal's right side, behind that spine, there's this lump of skin that looks like an old catcher's mitt. And that's because it kind of is. It's the preserved forefoot, the preserved paw. And what you're seeing is the underside, the skin on the palm of the hand, basically, that's preserved there. And there's also this large mass, about a soccer ball-sized mass, between the hips that we think is stomach contents. And we're currently analyzing, trying to figure out what this animal may have eaten. So all of these things kind of set it apart as a unique specimen. What I want to do is, even though it's very well preserved, it's not complete. It was hit by the excavator. Initial parts of it had knocked off or been removed before it was noticed. So even though it's incredibly well preserved, we only have about the first 2/3 of the animal. I'm going to zoom in on some of the osteoderms in the neck region and show you a bit more of the anatomy. Up top you have just the photograph, and on the bottom you have a bit of interpretation there. And the big thing here is that there's alternating rows of polygonal scales in this green color, and then osteoderms in an orange color. Remember, osteoderms are the bony components of the armor. But if you look closely, on top of the osteoderms, you see these dark gray patches that cover a lot of the osteoderms. What those represent is the epiosteodermal scale, or the keratinous scale, that would have once sat atop of the osteoderms. Just to give you a better context of this, let's go back to our extant comparison. So here's our crocodile. And if we look on the images on the left, that's what it looks like in life. It has this kind of leathery look to it, and the osteoderm is inside the skin. When all that skin is removed, and we just have the naked osteoderms, it looks like the condition on the right, where we have these kind of round, really porous, almost honeycomb-like bone structures. And that naked osteoderm is how we usually find them for ankylosaurs, whether they be isolated or associated with the skeleton. That's what we're used to seeing. But in this case, we don't just have that. We have the covering on top. And this is nicely shown by this great histological section by Burns and colleagues from 2011. So what they did is they took crocodile skin, or alligator skin, and cut a very thin section, looked at it under the microscope to identify the different tissues. Just very basically, we can talk about those three main tissues. We have the osteoderm itself, which is this triangular structure in the middle, highlighted in orange. That is held in a matrix of the dermis, the deep part of the skin, shown in green. And then on top of that osteoderm, you have this keratinous scale, which is epidermis. And that's that leathery or maybe even like fingernail type material that you have that's really tough on top of the osteoderm. So if we go back to our nodosaur, that's what we see preserved on those osteoderms. That's amazing. But we don't just have that on the neck. We have that throughout the whole animal. Here's osteoderms along the back, and you can see, again, rows of osteoderms and scales. And then there's that keratinous covering that's preserved on those osteoderms. And this is best seen on actually that shoulder spine. Here's a close-up view, and you can see the only part that is bone is that little orange bit there. The rest of the structure is all keratinous. It's all that horn-like material, that fingernail-like material that's preserved. So because we have these different tissues preserved, one of the first steps we have to do is actually map this out and show what's actually preserved in this animal. And we can do that. And in this case, the light yellow is the osteoderm, the bone of the osteoderm itself. And the dark gray on top is that keratinous scale. We don't usually have that. There's almost no other cases where that's preserved. You might notice in the sacral region there's none of that preserved. And that's because that's actually not prepared out in 3 dimensions. That was a part and counterpart. So it broke in the plane of the skin, almost like the pages of a book. So, one of the things that we can do to help identify these different types of tissues is look at it under different spectra of light. So this is what it looks like under visible light. So this is the light that you see with human eyes. And you can see different tissues that are fairly obvious. You can see this orange is the osteoderm, the black is the skin. We have some of the internal skeleton there. But what happens if we look at it under different wavelengths of light? So first off, we're going to look at it under UV or ultraviolet light. And it looks similar, but there are some things that jump out. First of all, there's these dark patches. Some of the osteoderms are darker. And that's because they were covered with glue in the field. And the glue actually acts to absorb that UV light. I want to stress that this isn't UV fluorescence. This isn't stuff that's being sent back in visible light. This is actually UV light that's reflected and captured with a UV modified capturing camera. And you can see here's a picture of Darren, I think that's Darren, applying glue to the osteoderms in the field to consolidate them. So using this, we can tell where glue has been applied and where we haven't had glue applied. You might also notice that there's this white patch in the top left, and that corresponds to the stomach contents there. So there's something there that's actually reflecting UV light more than everything else. What's really useful, though, is if we look at this under infrared light. Here you can see the osteoderms glow. They're reflecting a lot of that UV light. So they're really easy to identify. And that's in contrast to the endochondral bone, or the internal skeleton, which is kind of this medium gray color. It doesn't reflect the UV light nearly as much as the osteoderms. And then the black is this really UV or IR non-reflectant skin. So what we can do is we can combine these different wavelengths into what's called a false color composite. And all of this stuff was done with the help of Corey Jakolski, who helped with the multispectral photography. And really, this just helps you show the different types of tissues that are preserved that we don't normally get to see, or that aren't as distinct with our normal visible light spectrum that we're used to seeing. And this is useful because we can use this then to figure out blocks that are less obvious. So here's a block through the mid part of the animal. This bit here is that catcher's mitt of that front paw. And you can see that there's some interesting stuff happening in the middle here, but we're not really sure what it is. So if we look at it under ultraviolet, that's not overly helpful. But here's the infrared, and you can see these light up again. These are osteoderms. We can make that false color composite, And there's this very bright pink color. There's the osteoderms that are in situ or in place. And here's osteoderms that have actually been displaced as the body cavity collapsed after it was buried. You can contrast that with the internal skeleton, part of the hip there, preserved as the ilium. So techniques like this are actually very helpful, helping us map out the different tissues that are preserved. When we have a specimen like this, one of the first questions we need to ask is this something new? Does this represent a new species? And to answer that, we need to compare it to all the other known or named species and see, is it distinct from those, or is it already covered by one of those species? And really, we're looking for distinct or unique features. So we call these autapomorphies, or things that make this animal different. And what I'm going to do is I'm going to show you some of the features that make it unique or that are shared by a small number of animals. To do that, I'm going to use this beautiful skeletal illustration done by Donna Sloan. And just for reference, there's a googly eye in the orbit and a nostril there. So the top view is of the top of the skull looking down, and the one on the bottom is a side view looking at the skull and the first row of armor on the neck. One of the things that the armored dinosaurs have is they have a series of cranial dermal plates, also called capitegulae. And these are basically plates of bone that are either fused to the skull or outgrowths of the skull. And they cover most of the skull itself. And this animal has this one large kind of suboctagonal plate that's in the frontal parietal region, so kind of in the forehead region. But if you look in the nasal region, it has a whole bunch of small, round, irregular shaped plates. Above the eye, it has a supraorbital cranial dermal plate that has this very sharp ridge along the edge of it. And then below the eye, it has this very large jugal or cheek plate that's actually bigger than the eye socket or the orbit, and it has a sharp point on it as well. There's also a couple of features of the postcranial skeleton that jump out as being distinct. First off, I have to point out that it has these alternating rows of osteoderms and scales all the way down the body. And the rows of osteoderms don't come into contact, they're separated by those scales. The shoulder spine, that parascapular spine, is massive. It's the largest spine. It's about 1/2 meter long. It projects from the side of the body and then it curves backwards. The 3rd, 6th, and possibly 9th row on the back of these osteoderms, they kind of pinch out laterally. They don't make it to the side. That's a unique thing we don't really see in a lot of other ankylosaurs. And then finally, looking at the neck, the osteoderms in the cervical region, or the neck region, they have very prominent keels, and the apices of those keels project past the footprint of the osteoderm. So all of these things, and other things as well, suggest that this was a distinct new animal. So we gave it a new scientific name, and that's Borealopelta markmitchellii. Just to kind of explain this a little bit, the Borealopelta, boreal means northern in Latin. You hear it with boreal forest or aurora borealis. Pelta is from Greek. It means shield, and that's a common ending for armored dinosaur names. And then Mark Mitchell, of course, honors Mark Mitchell, the preparator who spent so much time working on it, and we felt that was a valuable contribution that should be rewarded with being named. So we know it's something distinct, but we don't really know who it's related to, which other animals it's related to. And that's the next question. And to do that, we look for shared features that unite a smaller group of animals. These are called synapomorphies. They're basically called shared derived characters. But there's a bit of a problem with this. And that's because, as I mentioned before, there's skin preserved all over the surface of the animal. So we can't actually see the internal skeleton. Now, most other ankylosaurs, we only know from the internal skeleton. So it's hard to make direct comparisons. You're like comparing apples to oranges. So ideally, it would be great to X-ray and see what's inside and see if we can demarcate the internal skeleton for comparison. And often we can do that using CT scanning. So we took it to Calgary and got it CT scanned. Unfortunately, this was not successful for a couple of reasons. The first reason was that the rock itself is radio opaque. Basically, it reflects back those X-rays. It doesn't allow them to penetrate. And just the size of the specimen and the amount of rock on it as well made it very hard to penetrate. So this didn't pan out as we were hoping. We are still working on trying to get it scanned with a more high-powered scanner. The problem is that the specimen is so large and so heavy, yet at the same time it's so delicate. It's difficult to transport, and it's also currently on display. So it's a balance between conserving the specimen, making it available for the public, and research. What we do is we look for a variety of characters or traits. And in this case, we're using the character matrix of Arbour et al. 2016. So a series of anatomical features that we quantify for each animal. And using this matrix, there's 92 for the skull, there's 66 for the postcranial skeleton, and there's 25 for the armor, so 183 in total. When we look at our animal, though, unfortunately, we can only code about half of those for the skull and half of those for the armor. That's because we can't see inside the skull, and it's also because we've lost the tail. The really bad news is we can only code about 8% for the internal skeleton, again, because it's cloaked, it's hidden by the skin. So all in all, we can only evaluate about 1/3 of these characters for this specimen, even though it's one of the best preserved in the world. And that's because it's exceptionally preserved. We have stuff that's more important that's actually blocking the internal skeleton. So in some ways, it's actually too well preserved. What we do once we've coded all these characters is we plug this into a computer program, and it makes all the possible trees, all the possible relationships between the different animals, and it evaluates how many evolutionary steps were needed to make that tree. And basically, it chooses the simplest tree, and unless we have other knowledge, we like the simplest answer. And as we add more data, we'll of course get different trees, and they'll become more complex, and they'll continually change. So this is the result from that analysis. This is a phylogeny, basically a family tree of armored dinosaurs. And it's also time calibrated. So the vertical axis here is time. Blue at the bottom is the Jurassic. Green kind of halfway up is the Cretaceous. And the way you read the pattern of relationships is that the closer the animals are in terms of the branching, the less steps you have to go through the tree, the more closely related they are. And we have the two distinct groups. We have Ankylosauridae and Nodosauridae. Unsurprisingly, our specimen comes out as being a nodosaur. So from now on, I'm going to be mainly talking about nodosaurids and not talking about Ankylosauridae at all. And it also occurs temporally about halfway through the range at which nodosaurs occur, and it occurs right in the middle of that group, which is interesting. So we can ask questions about what does this actually tell us about nodosaur evolution? Does it actually reveal anything interesting? And there's one thing that I think is quite interesting in that the earliest forms, things like Gargoylosaurus here from the Jurassic, if you look at those cranial dermal plates, they have a large number of irregular plates on the top of the skull. The latest forms, the forms we find in Dinosaur Provincial Park in the late Cretaceous here, things like Panopliosaurus edmontonia, they have a much reduced number of plates, and they're very complex, and they have very specific shapes. If we look at the things that Borealopelta comes out next to in terms of its closest relatives, those are Papasaurus and Uropelta, both from about the same time. But Papasaurus has a situation kind of like Gargoylosaurus, a very large number of irregular plates, whereas Uropelta looks very similar to those in the late Cretaceous. So what does Borealopelta look like? Well, I'll remind you, it has this one large plate over the forehead area, and then a whole bunch of small plates on the nasal. It's actually kind of transitional between these two forms. It looks like the back end of the skull, the capitegulae either fused together or reduced in number first, and that shifts anteriorly. That's just one of the things that this can reveal about the nodosaur relationships. But I think the coolest thing, the most exciting stuff, is actually what it can tell you that's unique, that other specimens can't tell you. And that's, of course, because of the preserved keratinous sheaths. So this is that same family tree. I've just rotated 90 degrees. And here are the 5 best preserved ankylosaurs. Again, light yellow are the osteoderms, light gray is skin or scales, and then the black are the coverings of the osteoderms. And the take-home message here is that the nodosaur that we have here is really the only one that preserves those keratinous scales in any number that we can actually analyze. So that's the cool thing that we can play around with and try to get some interesting answers for. So if we go back to our kind of tissue map of where the different tissues are preserved, one of the first things we have to do is actually kind of come up with a grid or a nomenclatural scheme to identify the different osteoderms. They have to come up with a kind of a numbering system. And we do that by basically dividing it into regions. So the neck or the cervical region is in red. The back or the thoracic region is in green. You have this transitional zone shown in orange. And then the sacrum or the hip area is in purple, and the forelimbs are in blue. And what we're going to be doing is, what we did was basically measure up all the different dimensions of these osteoderms, quantifying both the shape and the size, and seeing how that varies along the body, and seeing if there's interesting patterns. And really what we're interested in is something called allometry, which is how different measures scale relative to each other. So as one feature grows, does the other feature grow at the same rate, or does it grow faster or slower? And in particular, we're looking at how the height of those osteoderms, or how the overall length of those osteoderms changes. So this is a plot of the length of the base of the osteoderm versus the total length. So how long is that, including the apex, when the keel is included? And what you can see is that there's a very strong linear relationship here. All the data fall on one line, and pretty close to that line, except for one. It doesn't really matter which part of the body that they're from. So green, red, and yellow all fall along this line, except for this star. This is one outlier. It doesn't fit the pattern. This is that parascapular spine, or that shoulder spine. This plot is when we treat lefts and rights, and we average the two together. This is the same plot when we separate those and use them as two different data sets. There we go. And it shows they fall out right next to each other. So the left and the right are preserving the same pattern in terms of that shoulder spine is doing something different. What this means is that the osteoderms, although there's a huge variety in terms of the shape and the size from the neck to the back, most of that shape change can be explained by a simple mathematical equation showing how one shape changes relative to another shape. But that one mathematical equation does not explain the parascapular spine. Something different is happening with this spine. It's much taller than it should be compared to everything else. So why is that? Well, we can look at the specimen itself, and one of the things that we can see is that it has this really large keratinous structure. We saw that originally. Here's the osteoderm core, and this is all preserved keratinous sheath. This looks very different to the alligator that I showed you before with the histological section, and it actually looks superficially very much like what we'd see in actually a horn of something like a cow that's alive today, where you have, again, a bony core and this sheath. So horns, just like osteoderms, they have a bony core, and then that's covered in this keratinous tissue. In this case, it's called a sheath and not a scale. And it's often that keratinous covering that adds either interesting shapes or exaggerates the underlying size or shape of the horn core. So we can compare that large osteoderm in Borealopelta to the horns of modern mammals. And what you're seeing is basically the height of the yellow bar is the core, the bony part, and the height of the gray part is the keratinous part. You can see it compares very favorably to animals like bos and bison, the domestic cattle and bison that we have here today. And it also compares, in a relative sense, very favorably to horns of lizards today, especially chameleons. They're much smaller, but the relative size of the horn and the sheath is a much better analog than it is to something like a crocodile osteoderm. But we don't just have that one large spine preserved. We have the spines preserved from all over the body. We have all those keratinous coverings. So we can actually look at how that scales across the animal. So here's a similar allometric plot, but in this case, it's two different tissues. So on the x-axis here, we have the length of the bony core, so the yellow part, and on the y-axis, we have the length of the sheath. And we're looking at how those change size, or how they change proportion as the osteoderms get larger. And the big thing to highlight is that isometry, this dotted line here, that's the line where there's no shape change, where they grow at the same rate. The slope here is about 2.2 to 2.6, depending on the type of regression you use. That means that as the bony part grows at one rate, the keratinous sheath grows at a rate that's about 2.2 times or 2.5 times faster. So it's more than doubling the size as the osteoderms get larger. We can just visually see that. Here's some of those osteoderms. And for the small ones on the back, you can see about 3% or 6% of the total length is from that sheath. And that's similar to what we see in osteoderms of animals today, like an alligator. But when we have the large spine, about 25%, about a quarter of the entire length is that sheath. And that's more similar to what we see in a horn. But we can actually quantify this over a larger scale. So people have measured horns from a variety of living animals and published the data, so we can actually use that data and compare it directly to the nodosaur. This is that same plot I showed you before. Here's the nodosaur osteoderms. Here's that line of isometry. This is the bone part. This is the keratinous part. And what I'm going to do is I'm going to throw up data for a variety of living animals with horns. First off, here's the regression for the line of best fit, basically regression for the nodosaur. Here's the data for Phrenosoma. This is the horned lizard. We have these in southern Alberta. This is chameleons, the horned chameleons. Here are mountain goats, again, Alberta species. Pronghorns. And then we have our domestic cattle and our bison, bighorn sheep, and finally capra, some of these goats with these massive horns. I don't want you to take away all the details. I want you to take away two main results from this. The first is that these lines are not parallel. As the osteoderms get larger, these lines converge on each other. So as the osteoderms get larger, they become more horn-like. And the second thing is that the parascapular spine, shown with this green star here, falls right in the middle of the data for living domestic cattle in terms of the relative size of its horn and its sheath. So what this means is that when we compare extinct animals and try to figure out their behavior, we really want to compare them to modern animals. They're called analogs. And for most of the osteoderms, we've been comparing historically to things like crocodiles, and that makes a very good analog, and we think these might be for some sort of defense. I would argue that when we see really large, exaggerated osteoderms like the parascapular spine here, perhaps a better analog is something like the cranial horns of mammals or lizards today. Now, why does that matter? Well, if you look at the function of horns today, often you think of them as being defensive structures. And when push comes to shove, animals with horns will use them to defend themselves. You can see that here. But the evolutionary driver that resulted in the evolution and the exaggeration of those horns is, in almost all cases, some form of sexual display, whether it be combat with members of the same sex. So males competing over territory, or females, or males trying to impress females. The second thing I want to talk about in terms of the research is the preservation of color. And if I was giving this talk 10 years ago, I would have said, we don't know anything about dinosaur color. You can use your imagination. Artists can do whatever they want. That's changed in the last 10 years, mainly based on work from Jakob Winther, who's one of our co-authors on this particular project, looking at preserved color. And just a very quick explanation is that color can be preserved in fossils in two forms. It can be preserved chemically, so the actual original chemical structure is preserved, or it can be preserved structurally. For the melanin group of pigments, there's two types. There's eumelanin, which is the black pigment, so if you have black hair, that's mainly what's in your hair. And there's pheomelanin, which is the red color, so if you have red hair, it's mainly pheomelanin. And they have a different chemical structure, and they also have a different shape to the melanosomes. So melanosomes are basically little bundles containing pigment, and the shapes of those bundles depends on how much pheomelanin versus how much eumelanin they have in the bundle. And you can see here, here's an example comparing fresh melanosomes to fossil melanosomes. So we can infer color based on both of these things. So we invited Jakob to come and take some samples and see if he could find any melanosomes preserved. We have skin preserved, so maybe there's color preserved. The bottom SEM here, the scanning electron microscope image, shows what the texture looks like, and there's no melanosomes. Basically, either the heat or the pressure resulted in the melanosomes basically dissolving. So we don't know what's there. So that was a negative result. But we might still have some preserved in terms of the geochemistry. So that was the next question. And we, again, took samples, and we subjected those samples to mass spectrometry. Basically, small bits of the sample is destroyed, and then you look at the particles that come off of that, the charge and the mass of those, and you can figure out what the original organic compounds were. So in this case, this is just 4 samples. We've done lots of samples. But the top 3 are all samples from the skin, and the bottom sample is a control sample, so it's from the sediment. And each of those peaks represents a different organic molecule. And the ones highlighted in black are breakdown products of eumelanin, the black pigment, but they're not exclusive to eumelanin. And the ones in brown are eumelanin and pheomelanin. The really important one to highlight is the one, number 14 in red. That's an organic molecule called benzothiazole, and that's diagnostic to pheomelanin. So when pheomelanin breaks down due to heat or pressure, it produces benzothiazole. So based on this, we were able to suggest that the animal had contributions of both pheomelanin and eumelanin to its coloration. It likely had a rusty red color in life. Not only did we have the idea of what the coloration was, we could also look at the distribution of that color. And based on the cross-sections of the animal in several areas, you can see that those dark organic layers are much thicker on the back of the animal, and they petered out towards the sides. And this is most easily interpreted as reflecting the original pigmentation of the animal, so being dark on top and lighter underneath. This is a pattern called countershading. So why is this important? Well, this is the audience participation part of the talk. So there's 2 choices, 2 balls. One of them may appear to be coming out towards you, and one of them may not. So who thinks A appears to be coming towards you? And who thinks B appears to be coming towards you? Okay, you guys all passed. Do you know why you did that? Light source. So you made, whether you knew it or not, you made 2 assumptions. The first assumption you made was that the light was coming from above. And that means the top of the sphere is lit and the underside is in its own shadow. And the second assumption you made was that the object is of consistent color. Okay? And our eyes are attuned to pick this up as a 3-dimensional shape, even though I can assure you that screen is 2-dimensional. It's not just us. Other animals that are visual predators will rely on picking up these visual signals to see 3-dimensional objects. So if you're an animal and you want to hide, want to camouflage yourself, you have no control over where the sun is. That's beyond your evolutionary control. But you can control, in an evolutionary sense, your coloration or your pattern of coloration. And what many animals do is they have a pigment that's opposite the pattern of light. So dark on top, light underneath. When you add that to the pattern of the light source, they cancel each other out, and it helps to at least diminish your 3-dimensional outline. This is called a countershading or self-shadow concealment, and it's very common across pretty much all animals today. And there's some exceptions. One of the biggest exceptions is that when animals get large, they tend not to be countershaded anymore. Things like elephants and rhinos aren't countershaded. And things that get large as adults, they also tend not to be countershaded as adults. There's a great example with bison here. You can see the juvenile is countershaded. The adult's actually opposite countershaded. And this is because as adults, these animals do not face large predation pressures. So they don't need to hide on the landscape, whereas it's very important for smaller animals or for juveniles. And we can quantify this pattern looking at the number of species that are countershaded in different size classes. So getting larger towards the right, and you can see the percentage of countershaded taxa drops off to nothing. And that hashed area represents body sizes where animals are no longer subject to predation pressure as adults. So what does this matter for Borealopelta? Well, if we toss Borealopelta in there, it falls in that area where animals today are not countershaded and are not subject to predation. So what this means is that even though Borealopelta was a large 1.5-ton, heavily armored dinosaur, it still relied on visual camouflage to hide. It was still subject to predation pressure from the large carnivores of that day. And it just provides evidence to how much nastier the predators were back in the Cretaceous than they are now, and how much larger they were. So based on the preserved anatomy, the osteoderms, all the details of the scales, details of the pigment and the countershading, we were able to work with several different paleo artists to reconstruct what this animal looked like. And what you're seeing is 3 different visions of what this animal may have looked like back in the Cretaceous. And the bottom one really highlights this idea that even though this was a 1.5-ton dinosaur tank, it was still subject to predation. And one of the big predators at that time would have been something called Acrocanthosaurus. This large, it looks like Tyrannosaurus, but it's not actually very closely related to Tyrannosaurus at all. I do have a whole bunch of people to thank, and I'm sure there's people at the museum that I've forgotten off this list here.